Then if we look in families, if one sibling has autism, the risk for the next sibling is about 10%, plus or minus. This translates to a 20 to 50-fold increase over the general population risk, just depending on how you define it. So again, this is extremely high. Diseases like schizophrenia, that relative risk is 2. Bipolar disorder, dyslexia, 6. So this is huge. However, high heritability doesn't mean the genetics is going to be simple. The mode of inheritance, how that's passed down, is it like Huntington's disease, where it's a dominant gene, that if the parents have it, and you get it, you're going to have the disease? Or is it more complex than that, where the genetics is kind of a risk factor that just gives you an increased risk? So most models predict an interaction of 3 to 10 genes in any given patient. And this is consistent with things like diabetes, high blood pressure, heart disease, et cetera, where it's complex genetics. It's multiple genes interacting. Each of them is a risk factor that boosts the risk. And so many genes act together in these complex diseases. Common genetic variants may contribute. What that means is that the parents have the genetic variants, but they don't have the disorder. And the reason is because they don't have all of the variants. It comes from both of the parents. So if one has 2 variants and the other has 2, that mixes together to have 4, and you need all 4. Variable penetrance, and this basically means that, again, you can have one of the genetic risk factors and not show the condition. There are other things that interact, even chance. And variable expressivity, that 2 children with the same genetic mutation may look different. One may have severe language delay. The other may be severely mentally retarded. One may have seizures. The other may not. So there's variability. There's not a one-to-one correspondence between a genetic mutation. It just increases the risk. So knowing this, that it's highly genetic, people like me and a number of people around the world have spent the last 8 years or so working very hard to find genes. And in the last 2 or 3 years, we've had some remarkable successes that I'm going to talk about. And first, I'm going to tell you how we do this. So there are 3 basic kinds of studies that we do. One is we can look and find children who have an obvious chromosome anomaly or genetic syndrome, like a Down syndrome or Fragile X, those kind of things, where in Down syndrome there's an extra chromosome 21. We do genetic linkage and we do association. And I'm going to explain those to you very briefly, but I hope that this makes sense. Every human being has 23 pairs of chromosomes, one from mom, one from dad for each of those, plus what we call the sex chromosomes. If you're a woman, 2 X's. If you're a man, an X and a Y. And so in every cell, you have this complement of DNA. And that's where all your genes are. And what can happen is that for something like Down syndrome, you have an extra copy of one, an entire chromosome, an extra copy of one of these. Sometimes you might have a small piece deleted or an extra copy of a small piece. It doesn't have to be a whole chromosome. And this is called a structural chromosomal abnormality. It's a chromosomal disorder where it's broken or doubled. There's a term that you're going to hear because it's actually going to start coming out in medical parlance. It's a new discovery over the last 3 or 4 years, but it's going to come out into practice very shortly, and that's why I mention it here. When these structural variations are more than 1,000 base pairs, and your whole genome is 3 billion, so 1,000 is pretty small. You can't see that under a microscope. They're called copy number variation. That is, you have, instead of having that 2 normal copies, you have at that particular region 1 if you have a deletion, or 3 or 4 if you have extra copies. It's called a copy number variation. And all of us here have these, an average of 12 at least in our genome. And 5 years ago, we did not even know that. We thought that the source of all human variation was single base pair mutations. So I'm going to discuss that in a minute. And this becomes very important because these kind of things are a very significant contributor to autism. So I'm going to show you in a little bit later exactly how we find these. But new methods have allowed us to look at the chromosomes with great detail. Linkage analysis. I'm going to simplify this for you. We simply ask the question in a family, and you can see here are 4 individuals from a family with a shared disease region, and they have a great-great-great-grandparent, let's say, up there. There's genetic recombination that goes around every time they mate and have children, and we have people who have a disease, and they have a shared disease region that's pink up there. And within that region, the disease gene sits. And the only thing we are doing in linkage is asking the question, what region of the genome is associated with the disease in patients? So we go through asking very methodically, who has the disease, and do they have this particular region? Yes or no, throughout the whole chromosome. Genetic association, we're not looking in families for it, we're looking in populations. So we're basically asking, is there a kind of variation that is associated with disease? And you'll see this should become evident, hopefully, in this cartoon. So we might take patients, a large group of patients with autism and a large group of control subjects. And we'll look at their DNA, these genetic markers called single nucleotide polymorphisms. And it's essentially this. The DNA is like an alphabet, a genetic code, G, A, T, C, C. And if there's a change in any one of those letters, or what we call base pairs, that's a polymorphism. We have one between us every 300 to 1,000 base pairs. Some of them are just benign. They don't cause anything. Others cause diseases. So we measure those to find the ones that cause disease. And we might ask, if we're looking at 5 different of these single nucleotide polymorphisms, let's say this change there from a G to a T, we look through and we ask, what's the frequency in control? What's the frequency in autistic cases? Well, you can see number 1, 2, 3, it's about the same. But number 4, there's a big difference. It's much more frequent in autism. And we just do a statistical test to show that that's significant. And that's what association is. When you see more or less of a particular genetic form in a case of autism versus controls. That makes sense? So how have these methods really helped us to find genes, and what have we found? If you take 100 patients with autism and you look at their chromosomes under a microscope, it's very rare to find something that you can see under a microscope. The visible chromosomal anomalies, we call them. The Down syndrome and stuff like that. It's less than 1 in 200, 1 in 300 in cases of autism, just seeing something under a microscope. But advances in our technology now allow us to look at an unbelievably high resolution. So going back to this cartoon before, we can see things that are below the limit of even a microscope now. We can see things at the very submicroscopic level. And we do this with something called a microarray. And I bring that up because you're going to hear it. It's going to be in clinical practice and already is in many places. And basically, what you have along, that's just the genome is laid out from chromosome 1 to chromosome X and Y. And you can see there's just signal there. And it should be around 0. It should be flat. And the arrow is pointing to a place where there's a little blip, a little mountain there. And that means that there's an extra copy of that particular region. So what this technology allows us to do is scan through the entire genome in 1 or 2 days and ask at very high resolution, is there a gain or loss, a copy of any of the genes? And so there we have a duplication at 15q1113, which causes 1% of autism. And that's just that's one of the common causes. And so this is beginning to come into clinical use now. So what have we learned? And again, these are now called copy number variations, or CNV. So what do we know? Using that technology, we know that between 10 and 20%, and I'm just going to estimate, it depends on the population in the study, about 15% of people with autism have an identifiable genetic disorder based on this kind of technological screening. Fragile X syndrome is present in about 1%. Tuberous sclerosis is rare, but is always screened for. Rett syndrome, rare. Other genetic syndromes, again, these are rare syndromes, each of them, but they add up to about 1%. And then these recurrent chromosomal abnormalities, that is a piece of a loss or gain, a copy number variant. And these 3 each account for about 1% of autism. So 3%, those 3 alone, just their examples, 3%. And then you have things that are just rare breakages in the DNA, things that aren't recurrent, that we don't see at 1%, that we might see at 1 in 1,000. But 10 genes have been knocked out, and those are called de novo, they're new. And those are almost always, not all the time, causal. And those cause up to 10%. So what you're seeing is that autism now, what we know and what we've learned from genetics is that of 100 children that come in, we can diagnose today 15% with the etiology, which we couldn't do even 3 or 4 years ago. But that autism is a compilation of these 1%, each adding up. Does that make sense? So what do we do with that? How, from a research standpoint, is that disappointing? Is that discouraging? No, as a matter of fact. We can identify these today, and we can study them and use something called inverse mapping. Usually a geneticist looks at a disease like autism and tries to find the genes, to map the gene. We can do this inverse mapping where we already know the gene, because we can identify the 15%. And take all those patients with a 15q deletion or a 22q deletion or a Fragile X or a tuberous sclerosis, and we can ask, what do they share in common? What is it about their brain that makes them autistic? And study it by imaging, psychology and other things. And so there's a lot of hope now that instead of studying 10 or 15 patients for whom we don't know the etiology, they could have 15 different disorders. We can begin to study patients with the same form of autism and ask about treatment, trajectory, outcome, and all of that. And that's the hope for the future. So these rare chromosomal disorders and these single genetic syndromes have been extremely important and are going to turn out to tell us a lot about autism. But how about the rest? How about the other 80% that isn't caused by this, at least at this point? Well, we've used linkage and association, those two other methods, to find genes. These methods depend upon statistics, and to do good statistics, you need very large numbers. So we created a large resource with the Cure Autism Now Foundation, Now Autism Speaks, now over 1,000 families, called AGREE, the Autism Genetic Resource Exchange. And this has been used, this kind of resource that was created has brought over 140 new genetic laboratories into autism research that weren't in it before. Now, one of the issues, though, is that despite this huge sample, we haven't massively increased our power to detect autism genes. And one of the reasons is because of this incredible heterogeneity, the fact that it's not all one thing. There are a lot of factors that are contributing. And in fact, if we look at a map, this is a map of a chromosome, of all the chromosomes and where known autism genes are, what we call loci in the genome, places. It reflects an amazing amount of progress. You can see these red areas are areas of linkage. The little dots, the green and red dots, are areas of chromosome deletion or duplication. Almost every chromosome has an autism gene. And it's just remarkable what the map looks like. But it also tells us that, again, there are many, many different forms of autism.